Okay. Now, are you able to see my screen? Okay. So, I was stupid. I logged in with my Zoom account at UF, not my Santa Fe email, and so I wasn't actually the host. But we should be good now. So you're able to see the slide there? Okay. Okay. So, our lecture today is on chemistry. And I am one of the weird people that likes chemistry. Chemistry was actually the first class I taught at Saginaw Valley. I'm guessing a lot of you probably won't like chemistry, or at least you think you won't like chemistry, but I hope that I can change your mind about that. We're not going to go very in depth. We're going to just very, very briefly touch the surface. Lucky for you, I don't write the final exam. Remember, the final exam is written by other people in the department. The other people in the department don't like chemistry. And so this lecture is probably the least represented on the final exam. It doesn't mean it's not on there. It just means that there's not a ton of it on there. And so if you struggle with it just a little bit, particularly with the details, I wouldn't worry about it too much. But I would be happy to help you if you do struggle with it a bit. So chemistry is the study of both energy and matter. That is the definition of chemistry. Matter is something that has mass and takes up space. So everything around you has mass and takes up space. You know that we have three primary states of matter: solid, liquid, and gas, right? And so if you can look at something and say that is a solid, that is a liquid, or that is a gas, that is going to be matter. If you look at something and you say that is not a solid, a liquid, or a gas, most likely it is not matter. In that case, it is likely energy, because everything is matter or energy. And so it's a matter of figuring out which one you're dealing with. Energy is a lot harder to pin down. And energy comes in a lot of different forms. It looks very, very different. When you break it down, we define energy as the capacity or the ability to do work. And work is the ability to cause a change. If you can cause a change, that is doing work. And you have to use energy to cause that change to happen. Some forms of energy you're very familiar with, like heat and light. Those are both types of energy. Some types you might be familiar with are kinetic and potential energy. If you've ever taken physics, you certainly talked about kinetic and potential energy. Kinetic energy is the energy of movement. When something starts moving, it has kinetic energy. Potential energy is energy that something has because of its position. If I hold something up and I drop it, it's going to start moving. Even though I apply no force to it to make it move, I put energy into it when I lifted it up. Because I'm pulling it against the force of gravity, I have to put energy into it to get it further away from the ground. I'm putting energy into it in the form of potential energy, and then when I let go and it falls, that potential energy gets converted into kinetic energy. That's why it's called potential energy. It has the potential to become kinetic energy. Most of what we're going to talk about in this lecture here, in terms of energy, is going to be energy in the chemical form. Now think about when you burn wood or burn anything. You get energy out of that. And you're getting energy out of that in the form of heat, because fire is hot. You learned that when you were very, very young. And we get energy out of it in the form of light. We can see fire, right? We can see it with our eyes. That is light. Chemical energy is stored in chemical bonds. We're going to learn about chemical bonds today, and there's energy stored in them. If we break a chemical bond, almost always we will release chemical energy. And when you release chemical energy, it gets converted into another form, usually either heat or light. You've probably seen chemical reactions that when they happen, they get hot. Burning something is a chemical reaction. It's the reaction of wood with oxygen. That's all it is. It's a chemical reaction between wood and oxygen that's releasing heat and light when we break the bonds in that wood, the cellulose in that wood. Think of another example of a glow stick. If you've ever used a glow stick, in the little plastic tube, there's a glass tube. And when you bend it, you break the glass tube, causing the chemicals in the glass tube to mix with what's in the plastic tube on the outside. They react together, and they give off light. So that is chemical energy. There are lots and lots of other types of energy that we're not going to talk about here. They're not listed, but that is the little 3 dots there at the end of our slide. Energy comes in many, many forms that are not listed here. So when we talk about chemistry, we're going to start at the most basic level. And last week, when we looked at the levels of organization, we saw that the very top of that pyramid was atoms. And I said that atoms were the smallest part of an element. It is the smallest piece of something that you can have and have it still be that element. If you break it up any further, it is not that element anymore. At that point, it's what we call subatomic particles. Sub, smaller, or under atomic, so smaller than an atom. We'll see what those are in a slide or two. But an element is something that is made out of atoms. Those are our most basic building blocks. If you look at the periodic table of elements, what you're looking at is all of the different building blocks that we have to work with. Everything else is made out of those simple building blocks. There are different combinations of those building blocks. Now, we have a lot of different elements in our body, but 98.5% of our body weight is made up of only 4 elements. Hydrogen and oxygen, we're 2/3 water, right? And water is H2O, some hydrogen and some oxygen. So that's where a lot of that hydrogen and oxygen come from. Then the rest is mostly nitrogen and carbon. A lot of carbon. All of the stuff in our body that is not water and is not bone is going to be based off of a carbon backbone. And so you can probably guess the rest of that, the 1.5%, is a lot of calcium because of our bones, and our bones also have a lot of phosphorus. So we said that an atom was the most basic, the smallest unit of an element that we can get to. But I said that an atom can be broken apart into subatomic particles. And those subatomic particles are protons, neutrons, and electrons. A proton has a positive charge. Think P for proton, P for positive. And protons are found in the nucleus of an atom. So on the right here, we have a basic drawing of an element. This is probably something you've seen before. This is called the planetary model of an atom. In the middle, we have what's called the nucleus, and then flying around on the outside are what looks like these planets circling the sun. These planets are actually electrons. Electrons are down here at the bottom. They have a negative charge. So protons have a positive charge. Electrons have a negative charge. Now, we like to think about electrons flying around in orbits like this. But unfortunately, that is not how it actually works. It's a nice way to visualize it. But in reality, rather than orbits, what we have are clouds. So that's the bottom diagram. So we still have the nucleus in the middle with the positively charged protons. That's the red here. But then the electrons fly around in a cloud. It's not a nice, clean path. They move around and they're somewhere in this cloud. It's very nebulous, and that's on purpose. That's just the way it works. Now, if you notice, there's something else in the nucleus. And in this case, those are neutrons. Neutrons are neutral. They have no charge. Neutron, neutral, right? So we have protons that have positive charge, electrons that have a negative charge, and neutrons that have no charge. They are in the nucleus. Now, for you, in terms of how chemistry works, neutrons are going to play no role whatsoever. Neutrons are very, very boring until you start talking about nuclear chemistry, and then you're talking about a chemist and radiation and nuclear decay and all that stuff. Neutrons are going to play no role in anything we're going to learn today. Everything is about protons and electrons. Electrons are what cause chemical reactions. So here's a quick little question, just to make sure that you are keeping up with what's going on. So let me know what you think the right answer is. Yes. So the answer is A, protons. I don't know what, I'm trying to, my chat window disappeared. I'm not sure what's going on, but I heard somebody say A. Protons, P, start with a P, P for positive, all right? I think here I can get my chat window back. Oh yeah. Yeah, so a bunch of you had A. There we go. So what charge do electrons have? Electrons are negative, and neutrons have no charge. They're neutral. So now we can take our atoms and we can start combining them. When we combine at least two atoms together, we get what is called a molecule. And those atoms are going to be held together by a chemical bond. Now, I said, in order to be a molecule, all we have to do is have at least two atoms. They don't have to be different elements. We can have a molecule made up of two of the same element. But if the two atoms are different elements, we have a special name for that, and that is called a compound. A compound is a molecule that is made up of at least two different elements. H2O, water, is a compound. It has three atoms, two hydrogens and an oxygen, but there's hydrogen and there's oxygen. And so we have two different elements there, which makes it a compound. So just remember, a compound is a molecule that has at least two different elements. A molecule is at least two atoms, regardless of what element they're made of, that are held together by chemical bonds. Now we have multiple types of chemical bonds. These are the glue that are holding those atoms together in our molecules. We have ionic bonds, covalent bonds, which come in two flavors themselves, and hydrogen bonds. We're going to talk about each one. So here's a nice little table. We're going to go through this, but this is also very helpful in terms of studying. Right? There's lots of information here, but it's not all that complicated. So the first type of bond we're going to talk about is an ionic bond. Now in an ionic bond, it says this is caused by a transfer of electrons. When we transfer an electron, we're going to end up with two atoms that have a charge. Now here we go. So here we're looking at the process of forming an ionic bond. If we look at this, this is the process of making sodium chloride. Na is the symbol for sodium, and Cl is the symbol for chlorine. So this is the planetary model, right? In the middle we have our nucleus containing our protons, and then we have electrons flying around on the outside. Now protons had a positive charge, electrons had a negative charge, and an atom likes to have equal amounts of them. So if we have the same number of positives as we have negatives, the atom as a whole has no charge. But if we move an electron from one atom to another, we get charges. So in this situation, we see this sodium starts with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 electrons. Sodium, just so you know, also has 11 protons. 11 positives, 11 negatives, they cancel out, there's no charge. But if sodium loses an electron, if it gives one to chlorine, now it has 11 positives, but only 10 negatives. And so it has a positive 1 overall charge. It has an extra +1. Chlorine also started with an equal number of positives and negatives. But if it gains an electron, if it gets an electron from sodium, now it has an extra negative. And so chlorine is now negatively charged. So we have a positively charged sodium and a negatively charged chlorine. What do you know about positives and negatives? What do? They attract, right? You know positives and negatives attract. That is an ionic bond. We have a positive next to a negative. They're attracted to each other. That is an ionic bond. Another way of looking at this is sodium gives an electron to chlorine. But that's still sodium's electron. And sodium doesn't really trust chlorine with its electron. So it doesn't want to just leave it. It wants to keep an eye on chlorine. And so it is staying around, watching chlorine, staying right next to it so that chlorine can't leave with its electron. If I go back to our table, the next are our covalent bonds. A covalent bond is the sharing of electrons. Rather than an atom giving an electron to another, they're going to share it. So imagine a situation where you had two people standing next to each other. And one of them has a tennis ball in their hand. And they reach it out to the other person, and the other person grabs onto it. They are sharing that tennis ball. They are both holding on to the same tennis ball, and so they are stuck together. Wherever one person goes, the other person has to go. That is a covalent bond. And the tennis ball is an electron. They're both holding on to the same electron, and so they are stuck together. Now, I don't necessarily need you to know the difference between a nonpolar covalent and a polar covalent bond, but I definitely need you to know what a covalent bond is. A nonpolar covalent bond is when the two people holding on the tennis ball have the same strength. In this situation, each atom is pulling on the electron in equal amount, and for the most part, the only way that ever happens is when the two atoms sharing that electron are the same element. If you have two different elements sharing the same electron, one of them is going to be stronger than the other. It's just gonna happen. And so one of them holds that tennis ball just a little bit closer to them. And so it's not an equal sharing. The tennis ball is closer to me or it's closer to you. That's a polar covalent bond. I think I have... Nope, I guess I don't. Did I get ahead of myself? Yeah, I guess I did. So this is our polar covalent bond. This is water. This is oxygen and two hydrogens. It's H2O. And there's a polar covalent bond between the oxygen and the hydrogen. The oxygen and the hydrogen are sharing electrons, but they're not the same element, and so it is a polar covalent bond. Now, before we move on to the hydrogen bond, I forgot that I had this slide here. So remember, when we make an ionic bond, we make charged atoms that are positively charged and negatively charged. We have special names for those. A positively charged ion is called a cation. That's this here. A negatively charged ion is called an anion. There's a couple ways to remember which one is which. If you are familiar with electrical circuits or batteries or anything, the negative terminal on a battery is the anode, An. So an anion is the negatively charged ion. The way to remember cation is cations are positive. Think of a cat. I wish I normally have chemistry cat with that joke, and I forgot to put it in with these new slides. But cations are positive, and anions are negative, like an anode. We have a lot of these cations and anions in our body. In our inside of our cells, that is the intracellular fluid, the primary cation, the primary positively charged element in there is potassium, and the primary charged anion is phosphate. On the outside of our cells is sodium and chloride, salt water, right? On the outside of our cells is a lot of salt water. We have salt water inside of our cells too, but in order to make our cells work properly, we need a lot of potassium and phosphate. And we're going to see this when we start talking about our neurons and our muscles. There is a very, very, very good reason why we have a lot of potassium in our cells. The last type of bond is a hydrogen bond. A hydrogen bond is essentially the sharing of a hydrogen. I like that definition better than what our book has. Our book has attraction between partial positives and negatives, and to a chemist that's actually not correct. This is from our book, but it is not correct. A hydrogen bond is the sharing of a hydrogen. It's like a covalent bond with an electron, but instead of an electron, they're actually sharing an entire hydrogen atom. And that's what we're seeing here. This is our water molecules, and we have 3 of them. This hydrogen is halfway in between that oxygen and that oxygen. And so what ends up happening is these 2 oxygens share that hydrogen. And so when this oxygen gives up half of that hydrogen to this one, it's actually going to borrow half of a hydrogen from another water molecule, maybe up here. So in the end, every water has an oxygen and two hydrogens, but maybe it's actually 4 half hydrogens. But in the end, the water molecules are sharing a hydrogen. They're both holding on to it, and so again, they're stuck together. If they're both holding on to the same atom, they can't go anywhere. Now, hydrogen bonds are very, very important in life, and we're going to see that when we talk about DNA. And that's at the end of this lecture. So here's a quick review question for you. What type of bond shares electrons equally? Yes, it is C, the non-polar covalent bond. So there's 2 parts here. Which type of bond shares electrons? That's a covalent bond. Non-polar covalent bonds are equal sharing. Polar covalent bonds are unequal sharing. Hydrogen bonds share also, but they're sharing hydrogens rather than electrons. Here's another one. What type of bond was within or inside of a water molecule? So the key is within, inside of a water molecule. No. So go back, look at it. Within a water molecule, inside of a water molecule. Let's make it simple. Imagine you only had one water molecule, that there was nothing else around, just one single water molecule. What would be in there? That is a polar covalent bond. So that is between the oxygen and the hydrogen. And so if we only had one of these, this one wouldn't be around. There'd be nothing to share a hydrogen with. And so you can't have hydrogen bonding if there's nothing to share a hydrogen with. And so all we have left is this. And that was a polar covalent bond. Okay. Before we move on to reactions, are there any questions? No. Okay. So next thing we're going to talk about are chemical reactions. So we're going to take these atoms that we then put in the molecules, and we're going to start to do stuff with them. Chemical reactions, we're going to make stuff, we're going to break stuff down, we're going to blow stuff up, all the cool stuff. When we talk about chemical reactions in our body, we refer to that as metabolism. When you think of metabolism, you think of the breakdown of food, or you have a fast metabolism, a slow metabolism. It's how quickly you turn food into energy. Well, we convert food into energy through chemical reactions. That is your metabolism. It is the reactions that are happening inside of us. We have, in biology, 2 primary types of chemical reactions. We have synthesis reactions. Synthesis reactions take small things and make big things. And we have decomposition reactions. They take, I'll get back to that question in just a second. In decomposition reactions, we take big things and we break them down. Because biologists, actually they probably weren't biologists, they were probably physicians or medical people. They weren't happy taking the terms that chemists came up with that made sense. And so they had to come up with their own fancy words. And so in biology, in anatomy and physiology, we refer to synthesis reactions as anabolism. And we refer to decomposition reactions as catabolism. Now, I don't have a good way of remembering these other than the beginning of catabolism is cats. And cats destroy things. Decomposition reactions destroy things. Catabolism reactions destroy things. They break things down. That is literally how I remember this. If somebody asks me which one is which, I have to think, well, cats destroy things. Catabolism is decomposition. Because I originally learned this as synthesis and decomposition, and anabolism and catabolism were just unnecessary terms that got put on top of this. For the most part, when we make something, it requires energy. If you're going to build something, you have to put a lot of energy into that, right? It's never easy to make something. And when you break something down, you release energy. Now think of a decomposition or breaking down reaction that happens very, very, very quickly. That is an explosion. And when something explodes, it releases energy. You know that, right? You know when something explodes, it releases energy. So that is how I would suggest remembering which one requires energy. If you blow something up, you're certainly decomposing it, you're breaking it down, you're releasing energy. And so when you build something up, it must require energy. Now let me get back to the questions you just had on the last slide. So there was a question that said, so the polar was an O meaning. I'm not sure what that means. The polar was an O meaning. Yeah. Are you able to clarify that? Yeah, I can hear you. Go ahead. So when I was saying it was by itself, I was when you can see the slide I'm on right now, correct? So on this slide we have 3 water molecules. And when you have multiple water molecules, then you can get that hydrogen bonding. So I was just trying to simplify it and say, imagine we only had one of these water molecules here. There's only one type of bond present. If you only have one, you can't have hydrogen bonding. So the only thing that we can have is this polar covalent bond, which is between the oxygen and the hydrogen. And it's polar because they are different elements. Does that make sense? You sure? Okay. So then the next question. Yep. Yeah. Because the bond is between an H and an O, and they are different elements. Because they're different elements, it is automatically a polar covalent bond rather than a non-polar covalent bond. Yeah. So if we had a bond between the two H's, which we don't here, but if we did, that would be a non-polar covalent bond because they're the same element. Correct. Yep. So go ahead. Yeah. Correct. So we didn't talk too much about that. The reason it is covalent and not ionic, and this is not something I would ask you on an exam. This is not part of this course, but you're asking the question, so I'm going to give you your answer. It's because there is no metal involved. When we look at the periodic table, we divide things into metals and nonmetals. Metals are on the left part of the periodic table and nonmetals are on the right. If we have 2 nonmetals, it's a covalent bond. If we have a metal and a nonmetal, then it's an ionic bond. Because we have two nonmetals, we have no charges. There's no positive, there's no negative, there's nothing of that. And so it's a covalent bond rather than an ionic bond. Yep. The other question. Oh, I think that answered that other question. Did it? The person that wrote that, I don't want to say your name, but the person who wrote the second question there, did that answer your question? Oh, okay. Okay. Yeah, it seemed more like an explanation. Go ahead. Okay. Any other questions on this? I know it's complicated, but it's complicated for the people who write the exam also, and so hint, hint. Okay. Then again, the lecture exam I do write, and so there could be some in there, but I would not ask you a complicated question. Basically, if you know the difference between an ionic bond that transfers electrons and a covalent bond where we share electrons, you should be just fine. So here, just another example of the difference between a synthesis reaction and a decomposition reaction. A synthesis reaction, which was an anabolism reaction, we take two small things, put them together. In a decomposition reaction, we take one big thing and break it down into smaller things. Usually, we like to think of reactions as going in one direction, meaning we take these things, we react them together, and we get these things, and now we're done. But particularly in biology, reactions can go both directions. We can take these things, have a synthesis reaction to make this thing, but this thing can also break back apart in a decomposition reaction to make these two smaller things. And so we see that here. Now, we did not talk about chemical formulas or how to write a chemical reaction, so I don't expect you to understand what is going on here. But CO2, you probably know, is carbon dioxide. And you certainly know that H2O is water. So if we take carbon dioxide plus water, we can actually react those two things together to get this H2CO3. That is called carbonic acid. And carbonic acid, based on the way these arrows are going, notice we have an arrow going to the right saying we can take carbon dioxide and water and make it into carbonic acid, but we can also take carbonic acid and go backwards to make carbon dioxide and water. But we also have arrows on the right of carbonic acid saying we can take H2CO3, take one of the hydrogens off of it, make an H plus and HCO3. Now, you may not recognize HCO3 by its formula, but that is actually bicarbonate, like sodium bicarbonate. And where do you know sodium bicarbonate from? What is sodium bicarbonate? Anybody? That's baking soda. Yeah. Sodium bicarbonate is baking soda. And what does baking soda react with? Anybody? Vinegar, yeah. Make a volcano, right? Put the vinegar and the baking soda together, they react and they make a volcano. We're going to see in just a little bit, vinegar is an acid. This H plus here is like an acid. And so if we have bicarbonate and we add an acid, what we actually make is they come together, they go backwards. They go left here to make carbonic acid, which then goes further left to make CO2 and water. Now, what is CO2? We have three states of matter. Is carbon dioxide a solid, a liquid, or a gas? It's a gas, right? It's a greenhouse gas. When you mix vinegar and baking soda together, why does it make a foam? Because we had bubbles. Those bubbles are the carbon dioxide. And so you had a two-step reaction going on there. You put these two things together on the very right, which then converted to the thing in the middle, which then converted to the things on the left. This is a very important process in our body. We're going to learn about this. This is called a buffer. We couldn't live without this happening, and that's coming up possibly in just a few slides. Most of the reactions in our body are reversible. The only way we can make them irreversible, meaning it's a one and done, is that if one of the products, one of the things we make, disappears. So with our vinegar and our baking soda, if we make bubbles, and those bubbles come up and they float away, we can't take that carbon dioxide, combine it with water, and undo what we just did. The carbon dioxide is gone. There's no getting it back. So at that point, it is irreversible. You can't take the lava and put it back in the volcano. This happens a lot in our body. In that situation, when we mix the baking soda and the vinegar, that reaction happened very, very quickly. It's not something you're going to mix together and you're going to come back the next day and see if something happened. It happens very quickly. And there are three primary things that determine how quickly a reaction is going to happen. Number one is how concentrated your reactants are. Your reactants are the things you're mixing together that are going to react with each other. If you have concentrated vinegar and very concentrated baking soda and you mix it together, they're going to react very, very, very quickly. If you took your vinegar and you put just a few drops of it into a gallon of water and then poured that into your volcano, it's going to be a pretty sad volcano. It's not going to bubble up very much. It's going to be a very, very slow reaction because the vinegar is a very low concentration. Temperature also plays a role in reaction rate. For the most part, the hotter a reaction is, the faster it's going to go. We're going to see why in a later slide. But for right now, just remember, to get a reaction to happen, heat it up. That is why if you put something in the refrigerator, it goes bad slower. Because the reactions happen in the colder temperature slower. They're going to spoil slower. The last thing that affects reaction rates, and this is probably not something you're familiar with, is a catalyst. A catalyst is a third thing. Imagine we have two reactants that we're trying to react together, and we want them to react quickly. If we add a third thing, a catalyst, it will speed up the reaction. And a catalyst will always speed up the reaction. It is impossible for a catalyst to slow down a reaction. We'll talk more about the catalyst in a bit. The next thing we need to talk about are solutions. A solution is when one thing is dissolved in another. Now, because we are in a biology class and not a chemistry class, the solutions that we're going to talk about are always going to be water solutions. Things dissolved in water. When we talk about solutions, we have two other terms. We have solvents and solutes. The solvent is the thing that does the dissolving. Now we talk in normal everyday life, and you go to a hardware store, there's going to be a solvent aisle with mineral spirits and all kinds of different things that dissolve things. A solvent is the thing that does the dissolving. So in our body, the solvent is water, period. We are not going to have any other solvent in our body other than water. The solute is whatever is dissolved in that water. So imagine a situation where I have a glass of water. If I pour sugar into that glass of water and dissolve that sugar in the water, what is my solvent? The sugar or the water? Yes, The sugar is being dissolved into or by the water. So the water is the solvent. The sugar is the solute. Same thing if we're dealing with salt water. The salt is the solute. The water is the solvent. In our body, everything is dissolved in water. Water is very, very, very important. Life could not exist without water. Water is a very simple molecule at its most basic level. It's an oxygen and two hydrogens. But it does a lot of weird things that allow life to exist. And if it didn't have these weird properties, we could not be here talking to each other. Number one, water is a very, very, very good solvent. The fact that it is polar covalent allows it to dissolve a lot of different things. A non-polar covalent molecule will only be able to dissolve other non-polar things. An ionic solvent can only dissolve ionic things. But something with a polar covalent bond can dissolve both ionic and non-polar things. And so we like to talk about water as being the universal solvent. Now, water can't dissolve everything. We know that oil and water don't mix. Oil is just too non-polar. It's too different. But water is much better than basically anything else at dissolving a wide range of things, which allows us to have salt dissolved in our body while also having fat and a small amount of oil dissolved in our body. Another thing that makes water essential to life is something called cohesion. Remember that slide where we had the three water molecules and they were hydrogen bonded together. They are essentially connected together in a chain. That means that if we grab one of those water molecules and pull on it, it is going to pull the others behind it. Now, this isn't incredibly important in animal biology, in us, but in plant biology, in botany, it is. We have a heart that pumps blood throughout our body. Plants don't have a heart, yet plants need to be able to get water and nutrients from the soil up to the top of the plant. And so the way they do that is they allow water to evaporate out of the leaf. And as that water evaporates and it goes up, it pulls an entire chain of water molecules up with it. And at the bottom of that chain, it's down in the roots with the nutrients. And so as the water evaporates out the top of the plant, it pulls the water and the nutrients up from the roots into the rest of the plant. That is cohesion. Cohesion is things sticking to each other. That's different than adhesion. Think of an adhesive as like glue that you use to stick two different things together. Cohesion is two of the same things sticking together. The last thing that makes water so important is thermal stability. And by thermal stability, what we're saying is water resists temperature change. We exist in a world, in an atmosphere, an environment that changes temperature a lot. But we like to stay the same temperature. And so if I am a temperature and I am mostly water and I go outside where it is cold, I'm not going to cool off all that quickly. Whereas if I had a metal pot and I took that outside, it's going to cool down very, very quickly. We are in summer in Florida, and so if I took a metal pot outside and put it in the sun, within 30 seconds you're going to burn yourself by touching that metal pot. But me, made of water, staying right next to it, are barely going to change temperature in the same amount of time. This ability to prevent temperature fluctuations allow us to stay the same temperature that we want to be, even though the temperatures around us change. I'm going to skip this slide. This isn't as important as it was in my mind when I put it there. Another thing to talk about, acids and bases. You know about acids. We said that vinegar was an acid. Acids react with bases. And we said vinegar reacts with baking soda. Baking soda is a base. Another term for a base is an alkali. If we have a lot of acid, we say the situation or the solution is acidic. If we have a lot of base, we can either say it is basic or we can say it is alkaline. So take alkali and add an Ne on the end to make it alkaline. An acid donates H pluses. An H plus is a hydrogen ion. This is a hydrogen cation. A hydrogen atom only has one proton and one electron. And so if we get rid of an electron to get a positive charge, all we have left is a proton. And so in kind of chemistry slang, we refer to hydrogen ions as protons. We say that acids are proton donors. Acids give protons or hydrogen ions to a base. The base is the proton or hydrogen ion acceptor. Vinegar gives a hydrogen ion to a base. These are the definitions of acids and bases. Acids give hydrogen ions to bases. Bases accept hydrogen ions from protons. So here we see what happens when we dissolve an acid in water and a base in water. If we take an acid like vinegar or hydrochloric acid or sulfuric acid and we put it into water, we get hydrogen ions off of it. They just break off when they start floating around. If we take a base and we put it into a water, we get OH ions. These are called hydroxide ions. Now imagine a situation where we took this acid and this base and we reacted an H plus with an OH minus. What do we get? If you take H plus and add it to OH minus, what do you get? So H plus has one hydrogen. Yeah, there you go. We get water. H plus has one hydrogen. OH has an oxygen and a hydrogen. Add an H to it. We have two hydrogens and an oxygen. That is water. If one starts with a positive charge, one starts with a negative charge, plus one and minus one cancel out. We have no charge. And so we get water. Acids and bases react together to form water. That is what they do. When we talk about acids and bases, we refer to pH. And I'm sure you have heard of pH before. Some of you probably have a better understanding of it than others, but I'm guessing you've all heard of pH. pH is a measure of how many hydrogen ions we have floating around. Now remember, acids give us hydrogen ions. And so if we have an acid, we have a lot of hydrogen ions. If we have a base, we have a lot of OH ions, not a lot of hydrogen ions. And so that is basic. On the pH scale, 7 is neutral, meaning we have the same number of H's as OH's. It is neither acidic or basic. An acidic solution has a pH less than 7. It could be 6, it could be 1, it could be 0, it could be negative 100. It is still less than 7. That is acidic. That means that we have more acid than base. If we have more base than acid, our pH is going to be higher than 7. We say that we are basic or alkaline. On this slide, we see some things that you're familiar with, and it tells us the pH of them. So pure water is 7. Now, we don't have pure water. There's always going to be stuff dissolved in it. Even we get gases from the air dissolving in our water. So in real life, we are never, ever, ever going to have perfectly neutral, pure water. But in a theoretical world, pure water is 7. Our body, most of our body, our blood, most of our cells, have a pH of about 7.4. So we are just a little bit basic. Other things in our body, like urine and saliva, are a little bit acidic. We know that something like lemon juice or orange juice is acidic. There's a lot of citric acid in there, right? And so it is acidic. And so lemon juice, vinegar, they're about pH 2. Orange juice is 2.5, 3.5, somewhere in there. Coffee is 5. Battery acid, that's a strong acid. That is not something that's compatible with life, but it was something we can make in a lab. That's pH 0. As we start going up on the pH scale, salt water in the ocean is about 8. Baking soda is about 9.5. Ammonia, you get into 10, 11. Bleach is 13.5, which is very strong. And then if you have like Drano, Mr. Plumber, that you're pouring down your drain to get all the hair out of your pipes, which is not a good idea. Clean out the trap instead. That is pH 14. That is very, very, very strong base. Everybody okay with the pH scale? Say it again. Oh, lye is... Have you ever... Probably. So lye is sodium hydroxide. So that's the chemical name is sodium hydroxide. But we get lye when you burn wood in the ash. The ash of burned wood is very basic. You get sodium hydroxide out of that. It's in the ash. That is lye. Nowadays, if you have something that has lye in it, most likely it was made in a lab. It's not from ash. But in the old days, when they didn't have factories that made chemicals, they would use burned wood ash as a source of base. Yeah, it's a good question. So go ahead. Somebody else have a question? Okay. So we had acids. Acids gave us hydrogen ions. Bases gave us OH ions. But some ionic compounds, some things made of ionic bonds, don't give us H's or OH's. Like salt. Salt gives us sodium and chloride. That is salt. That is a salt. Salt is an example of a salt. Table salt is sodium chloride. But the chemistry term salt simply refers to any ionic compound that when we put it in water, it breaks apart into a positive and negative ion, neither of which is a hydrogen or an OH. So sodium chloride is an example. Calcium chloride is an example. Potassium bromide is an example. There's lots and lots and lots of them. Anytime that we put a salt in water and we get positive and negative ions, that is going to be capable of conducting electricity. Pure water does not conduct electricity. We have to have things dissolved in it. A water solution that is capable of conducting electricity is called an electrolyte. Now, our nervous system, we talked about last week, signals throughout our body using electrical signals. And so we have to have electrolytes in our body. Now, we hear about electrolytes in commercials and things. Where do we hear about electrolytes? Gatorade, yeah. I'm the first one to talk about and say how Gatorade is better than Powerade or Allsport, if you're old enough to remember Allsport. But they really pushed an electrolyte thing. And what did we just say is an example of a very, very common electrolyte? What is something that we put in water and breaks apart but doesn't give us a hydrogen or an OH? Salt. Yes. And so Gatorade is mostly salt water. The electrolytes that they're selling are salt. Yeah. Now, there's a reason it's in there. Because when we sweat, we lose salt, right? That is why it is in there. It's also in there because if we have the right concentration of salt in there, the same concentration of salt in what we drink as is in our body, our stomach and our intestines are going to absorb it faster. So that's a good way of getting water into our system quickly. But when they're pushing, you have to replace the electrolytes you lose when you're exercising. They're just selling you salt water. It's flavored salt water with some sugar in it. That's all it really is. Most likely, I imagine so. So there is a reason why they did a lot of testing to find the perfect salt concentration, because the goal of it is to get water into our body as quickly as possible. And so they put just the right amount of salt in there to match our body salt concentration so that it gets absorbed quickly. They also put just the right amount of sugar in there. We are not going to learn this semester, but one way we get water into our bodies, we have pumps in our intestines that pump the water from inside of our intestines into our body. And those pumps run on sugar. And so if we put sugar in the water also, we have the energy to run the pump to get the water into our body. And so that is the purpose of that. Now, it sounds very simple, and it is, but it's also very, very important. I heard once, and I was taught when I was in grad school, remember, Gatorade was invented in the physiology department at UF, and I was kind of partly in the physiology department, and I was taught this by someone in that department, and they claimed that sports drinks like Gatorade and Powerade have saved more lives than any other drug in the history of humankind. Now that seems like a huge claim. In the US, that's probably not true. But in other parts of the world where people suffer from diseases like cholera, there are a lot of people that die of dehydration simply because the disease that they have gives them very, very bad diarrhea. We're talking millions and millions of people that die from diseases like this. If you give the body enough time, it can fight off the disease. And so if you give them sports drinks, keep them hydrated, they will survive. It doesn't take some fancy medication. The immune system is very good, but you have to give the body enough time to fight it off. Okay. So along the lines of our salts and our pH, I referred to a buffer before. Remember when we talked about the carbon dioxide and the carbonic acid and the bicarbonate. I told you this was very, very important for maintaining the pH of our body. And when we looked at the pH scale, we saw that blood was about 7.4. Our blood wants to stay roughly between 7.35 and 7.45. And if we go outside of that range, we're going to get sick. If we go far outside of that range, we are going to die. The primary way that we keep ourselves in that range is using what's called the bicarbonate buffer. And the bicarbonate buffer is that little reaction we were looking at before, the three-step reaction. Our body makes bicarbonate. Our kidneys, one of the major functions of our kidneys is to make bicarbonate. When we break down sugars, when we make energy, our body makes acids. It's just a byproduct of that process. And if we didn't have bicarbonate sitting around, a base, to react with the acid we made, our body would very, 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 very quickly become acidic and we would die. And so we have what we call this buffer. Buffers resist changes in pH. That is the definition of a buffer. It resists changes in pH. So if we add an acid or we add a base, the pH barely changes. It helps us maintain homeostasis. It keeps us in our happy place. And the bicarbonate buffer is the primary buffer in our body that helps control that. So that is the end of our basic chemistry. Now I'm going to give you a little break, but before I give you the break, do you have any questions about very basic chemistry, the stuff that we've covered so far? Okay, so let's plan to come back in 5 minutes. Oop, somebody had a question. What exactly is bicarbonate? Let me go back. If you don't need the answer to this question, you go ahead and take a early break. I will plan to come back in 5 minutes once we're finished with questions. Where was it? Here we go. So bicarbonate is this HCO3 minus. Because it has a minus charge, it likes to react with something with a positive charge, which H plus does. And so that is why it's a base, it reacts with an acid. Bicarbonate is it's this little molecule. It is a hydrogen, a carbon, 3 oxygens with a negative charge. That is what bicarbonate is. Sodium bicarbonate is bicarbonate with a sodium. Rather than a hydrogen with a positive, it is sodium with a positive. And they don't actually react and then break apart like this. They just come together and they're happy little friends sitting next to each other as sodium bicarbonate. Other questions? Okay, so let's take a 5-minute break and we'll come back and we'll talk about organic chemistry and biochemistry. So we're going to take what we just learned and apply it more to life. Out of curiosity, did anyone try to watch the videos, the recorded video? Did it work? So I think it's probably been 5 minutes. People can come, if people aren't back yet, they can join us when they get back. They won't exactly bother us or anything. So we're going to get started. And like I said, we're going to talk about organic chemistry. And particularly, we're going to talk about biochemistry and how organic chemistry fits into biochemistry. Organic chemistry is bigger than bigger than biochemistry. Organic chemistry is the study of compounds containing carbon. Period. If there's carbon in it, that is organic chemistry. Now there is a lot of organic chemistry that is different than biochemistry. All of your plastics, all of that stuff, that's organic chemistry. That has nothing to do with biochemistry. But in our bodies, in life, we have 4 types of carbon compounds that we can't live without. We have carbohydrates, lipids, proteins, and nucleotides / nucleic acids. We're going to talk about each one of those on their own. So we're going to start with carbohydrates. Now, carbohydrates are sugars. When you think carbohydrate, think sugar. They're called carbohydrates because we, for the most part, have carbon and we have water. Carbohydrate. And they come in different sizes. We have what we call monosaccharides, disaccharides, and polysaccharides. Mono means one. Saccharide means sugar. So a monosaccharide is one sugar. This is the most simple carbohydrate that we can come up with. And there are quite a few different monosaccharides. But there are 3 that are important to us. Glucose, fructose, and galactose. Glucose, you've certainly heard of. Fructose, you've certainly heard of, right? That's the devil molecule. In fact, I can hear Mama Boucher right now saying fructose is the devil. It's actually not all that bad. It's when you have too much of it, it causes a problem. But too much of anything causes a problem. We'll see just how important fructose is, I think, on the next slide. The third important monosaccharide that you probably haven't heard of is galactose. We have the molecules down at the bottom. Now, again, this is not a chemistry course. You have not taken organic chemistry. You don't know what you're looking at here. And you don't need to know what you're looking at here. But it's interesting to look at. You can see that these are all ring structures, right? They're kind of circular. 2 of them are hexagons. 1 of them is a pentagon. But they're kind of they're kind of circular structure. That is what a sugar looks like. For the most part, it is circular. Sometimes we can draw it linear. It's actually kind of like a worm or snake that can bite its tail. And so it can be circular or it can be straight out in a line. But these are monosaccharides. They are one sugar. They are one kind of circle. If we take 2 of those monosaccharides and connect them together, we get a disaccharide. So we see 3 of them here. This is sucrose. We have this sugar attached to this sugar. And this is a glucose attached to a fructose. And this is sucrose. Does anybody know what sucrose is? It's another name for sucrose. Sugar, yeah. Just like salt is an example of a salt, sugar is an example of a sugar. Table sugar is sucrose. Table sugar is made up of a glucose and a fructose stuck together. That's all it is. Another example of a very important disaccharide is lactose. Where is lactose? Yes, milk. Milk has lactose. The third disaccharide that is very important, but you might not have heard of, is maltose. What does maltose remind you of? Anybody? Well, what does maltose, the word maltose, what does that bring to mind? A malt milkshake or a malted milk bar like a Whopper, right? So malted means there's maltose. We do something to it to get maltose. Maltose is just two glucoses stuck together. And this is important for our body because maltose is going to be a very, very good molecule for transporting glucose. Because it is two glucoses stuck together. The favorite sugar of our body to burn is glucose. So maltose is going to be a very convenient way of transporting that sugar around the body. Particularly, our brain has a very good appetite for maltose. Our neurons in our brain like to burn maltose. So if we take a whole bunch of monosaccharides and string them together, we get what's called a polysaccharide. Poly is many, saccharide is sugar. Now, for the most part, at least in our class and in life, polysaccharides are always made up of long chains of glucose. It's not going to be a mixture of glucose and fructose. It's not going to be a long string of galactose or anything like that. Polysaccharides are very long strings of glucose. And in life, we have a lot of examples of this. In our bodies, we are animals. In animals, we have glycogen. Glycogen is the storage form of glucose. We're not going to learn too much about this semester because this is something you'll learn next semester in 2086. But insulin, which you certainly know about, insulin takes the glucose out of our body, uses it to make glycogen, and then that glycogen gets stored in our liver. When our blood sugar starts dropping, we take that glycogen, we break it back apart into glucose, and that brings our blood glucose level back up. In plants, they don't have glycogen. They have something that's very, very, very similar called starch. Where do you know starch from? What plant do you know is starch? Say it again. Corn, yes. Corn, potatoes, basically the plants that taste good. At least the plant parts that taste good. If you're talking like salads and stuff like that, it doesn't taste good, it's not starch. The reason starches taste good is because they're made out of glucose. They're sugar. They taste good. When we start talking about the icky parts of the plant, that is made primarily of cellulose. Cellulose is another slightly different chain of glucose. It can't be digested. What is the term that we give to parts of our food that can't be digested? Fiber. Yes. And so when we eat plants, we get starch and we get fiber. There's not a whole lot else in there. Yet there are minerals and vitamins and things like that. But in terms of what we're eating, they make up a tiny little portion. There is starch, there is fiber, and there is water. And that's more or less it. But glycogen, starch, and cellulose are all just long chains of glucose. The only difference is how they're connected. Are they linear? Are they a long straight string? Are they branched? How do they come apart? So, what is the answer to this? We just talked about it. Yes, cellulose. Cellulose we cannot digest. Are there animals that can digest cellulose, or can all animals not digest cellulose? Yep. Cows, horses. Think of animals that eat plants. They can more or less digest cellulose. So, if they can, why do you think they can and we can't? What's different between us where they have to and we don't? So we only have one stomach. Why don't we have 7 stomachs or however many stomachs a cow has so that we can break down cellulose? What's different about what we eat versus what a cow eats? They eat just plants. Yes, they're herbivores. If they can't break down the cellulose, they're going to have to eat a whole bunch of plants to get the. There's not much starch in grass. It's almost all cellulose. Like somebody else said, we can eat meat. We are hunters and gatherers, right? Nowadays we hunt down the meat in the meat case at Publix. We gather the stuff off the aisles and the shelves at Publix. But we go out, we find what we need, and we eat it. If we need protein, we eat meat. If we need sugar, we eat sugar. We eat whatever we want. And so we don't have to devote so much space in our body to breaking down cellulose. We can just get that energy someplace else. Yeah, go ahead. So vegans have to be very careful about what they eat, because if they just eat lettuce, they're not going to live very long. So what is something that what's a type of plant that vegans eat a lot of? They need protein, right? Where do they get their protein? Anybody. Right, but does anybody know if a vegan or a vegetarian, if they need to get protein into their diet, where do they get it? Beans. Yeah. Beans have a lot of protein. Yeah. But cows don't have access to beans. And so they have to break down the cellulose. But someone who has a strictly plant-based diet has to be very careful, and they have to make sure that they're getting those nutrients that they can't get from things like lettuce. Correct. They have to make sure they're eating lots of different types of plants. Okay, so that was carbohydrates. The next type of organic molecule we learned about is lipids. Lipids are oils and fats. Okay, lipids are hydrophobic. Hydro refers to water. Phobic refers to fearing. Water-fearing molecules, meaning that they do not dissolve in water, which technically means they're insoluble in water. Something that is soluble in water can dissolve in water. Something that is insoluble in water cannot dissolve in water. And I said this is fats and oils, and you know that fats and oils do not dissolve in water. We have 4 primary types of lipids in our bodies. We'll talk about each one again. We have fatty acids, triglycerides, phospholipids, and steroids. You've probably heard of at least a couple of them, probably not all 4 though. These are fatty acids. Now, again, this is a chemical structure that I do not expect you to be able to look at and understand what you're looking at. But what I need to show you is the difference between a saturated fatty acid and an unsaturated fatty acid, which is something you have certainly heard about. You've heard of saturated fats and unsaturated fats, but you probably didn't know what the difference was between them. So when you look at the saturated fat on the top and unsaturated fat on the bottom, the difference between them is this red part right here. There's a double bond here between these 2 carbons, whereas in the saturated fat, it's all single bonds. When we put this double bond in here, we lost 2 hydrogens. Outside of chemistry, when we talk about something as saturated or saturated with water, we say that it can't hold any more water. If we have a paper towel or a sponge that is fully saturated, it can't hold any water. If the ground is saturated and it rains, we get a flood because the ground can't hold any more water. In fatty acids, saturated and unsaturated returns to the amount or refers to the number of hydrogens that we have. A saturated fat has all of the hydrogens it can hold. An unsaturated fat is missing hydrogens and has a double bond instead of single bonds. Unsaturated fats have slightly fewer calories in them in the end than saturated fats because, number one, to break this down, we have to put energy into this to turn it into a saturated fat. Our body breaks down saturated fats. We don't have a good way of breaking down unsaturated fat. So we have to convert an unsaturated fat into a saturated fat, and that takes energy. Triglycerides are a storage form of fatty acids. These were our fatty acids. Notice that they are long chains. If we want to store them as fat in our body, we store them in the form of a triglyceride. Do I have a picture? No, I don't. I wish I did. But a triglyceride is 3 fatty acids. That is the tri here. And then they are connected to a backbone. Can I draw? Is it going to let me? Is this going to work? Come on. My computer is doing so many things it doesn't like me trying to draw. Now I'm going to be lucky if I get it to advance to the next slide. Oh, here we go. Here we go. So imagine I had a backbone, and then I had 3 fatty acids coming off of it. That's fatty acid 1, 2, 3, and then we have a glycerin backbone. That is the triglyceride. 3 fatty acids and a glycerin gives us a triglyceride, and that is the storage form of fat in our body. If you look at a person and you see fat, you're seeing triglyceride. If you are eating a piece of meat and you see fat in it, you are seeing triglycerides. Fatty acids are only used for transporting fat from one location in the body to another, they are short-lived. You can't see them. The third type of lipid is a phospholipid. These are primarily in our cell membrane. We're going to talk about cells next week. But our cell is basically a water balloon. In the membrane on the outside of that cell is the cell membrane, and it is made out of phospholipids. A phospholipid has two primary parts. There is the lipid part, and then there is the phospho part. If we look at the diagram in the bottom right, we're looking at a phospholipid here. It has a phospho head, and then it has two lipid tails coming out of the bottom. Now, we said that lipids are hydrophobic. They do not dissolve in water. The phospho part, though, is hydrophilic. Philic means loving, water-loving. So the head wants to be around water. The tail wants to be away from water. But our body is mostly water. Our cells are full of water. Our cells are surrounded by water. And so what we get is this phospholipid bilayer that you see in the left part of the diagram. We actually get two layers of phospholipids. And in both situations, the tails point towards each other. What you're looking at here is a cell membrane. At the bottom, the cytoplasm here is the inside of the cell, which is water. The top, the extracellular space, is the water outside of the cell. So the tails want to get away from that water. And so they point away from the water and towards themselves, and the hydrophilic heads point towards the water, and we get this very, very unique double layer structure. This is called the phospholipid bilayer. This allows the hydrophobic parts to stay in the middle and the hydrophilic parts to touch the water. Now I'm going to ask you a kind of theoretical, philosophical question. What is the advantage if we are made of water and the lipid part doesn't like water? Why not just get rid of the lipid part? Why not just have the phospho part? Why do we have to have that lipid part? What would not work if we didn't? Exactly, yeah. If we have water outside the cell and water inside the cell, we have to have something separating the two. And if all we had was the hydrophilic phosphohead, it would just dissolve and float away. And so we have no way of, like she said, we have no way of compartmentalizing the inside of the cell from the outside of the cell. That's a very good way of putting it. The fourth type of lipid is steroids. Now, when you hear steroids, what do you think of? Bodybuilders, Barry Bonds, Mark McGuire, right? You think of bad steroids. Steroids are not inherently bad. We're full of steroids. We can't live without steroids. Steroids are a type of lipid, that's all they are, and they are signaling molecules. You know, remember last week, we talked about signaling molecules as part of the endocrine system. Steroids are hormones. All of the hormones in our body are steroids. And steroids are made from cholesterol. Cholesterol is a steroid. It itself is not a signaling molecule, but it is the starting point, the building block to make all of the steroids in our body. So other steroids include estrogen, testosterone, and a whole bunch of other things that we can't live without. Steroids get their bad name because people take advantage of them and they take extra steroids trying to, as basically as drugs, to influence our body. So that is the end of the lipids. We talked about carbohydrates, we talked about lipids. Now we move on to proteins. A protein is a chain of more than 50 amino acids. Now you're saying, what the heck is an amino acid? An amino acid is, let me bring all these up. An amino acid is the basic building block of a protein. We have X number, as soon as I saw this slide, I realized not positive, I think it's 23 amino acids. Somebody's going to Google it right now and correct me. But I think we have a certain number of different amino acids in our body that we are capable of stringing together to make proteins. And so we take them, we put them together, and we get a string, and that string is called a protein. Proteins make up most of the organic compounds in our body. Compared to carbohydrates, lipids, and nucleic acids, we have the most proteins. Protein are amino acids strung together and held together by something called a peptide bond. Now, I'm not going to teach you what a peptide bond is, but you need to know that amino acids in a protein are held together by peptide bonds. That's a very, very common question to ask on homeworks, exams, and final exams. So you need to know that a peptide bond holds amino acids together to make proteins. Peptide bonds are a special type of covalent bond. Okay? Particularly, they are polar covalent bonds, but they are a very, very specific type. When we string our amino acids together to make a protein, proteins have structure. The proteins in our body are there for a purpose. A protein has a purpose. It is not for storing energy. It is there to do something. It is either going to have an active function or it's going to be structural. In either case, it has a role to play. And it can only play that role if it has the right structure, the right shape to it. So when we talk about proteins, we refer to its structure, and the structure comes in primary, secondary, tertiary, and quaternary structure. So you need to know the difference between each level of structure. The primary structure is simply what the sequence of amino acids is. Is it proline, histidine, asparagine, tyrosine, serine? Those are amino acids. What is the order of the amino acids? That is the primary structure. Each amino acid has its own slightly different shape to it. And so if you continue to put curved amino acids in a line, you get a curved kind of helical structure. If you put straight amino acids in line, we get like a straight piece of spaghetti. And so that next level of shape is the secondary structure. And that can either be a helix, like we see here. It can be a sheet, like we see here, where it kind of goes straight and then makes a hairpin and comes back. And it's going like this, creating a sheet. Or it can just be a straight line, connecting different areas of the protein. Once we have that, different parts of the protein, maybe we make our protein and we have a helix over here and a sheet over here, but they like to be next to each other. And so the protein is going to fold to bring them next to each other. That folding within the protein is called the tertiary structure. Most proteins only have a primary, a secondary, and a tertiary structure. Some proteins, though, also have a quaternary structure. And these proteins are unique in that, in actuality, they involve multiple proteins that come together and function as one. The best example of this is hemoglobin. Hemoglobin is the protein in our red blood cells that carries oxygen. That is the red protein that makes our red blood cells red. A hemoglobin protein is actually 4 individual proteins that on their own can't serve their purpose. But when they come together, they are hemoglobin. And so they come together. If you're looking at this, we have 1, 2, 3, 4. That structure with 4 of them together is the quaternary structure. Quaternary refers to the 4th. Secondary, tertiary means 3rd, quaternary means 4th. Is everybody clear on protein structure? No? Is there something specific you don't understand? Okay, so that's protein. Okay. Yeah. So imagine making a protein out of Legos. Okay, and each type of Lego that you have, they come in different shapes and different sizes, right? Different colors. The order. Imagine putting one on top of another, on top of another, on top of another, on top of another. That the order that you put the different types of Legos in is the primary structure. If each individual Lego block has its own shape, as you put them in order, it's going to start to bend and give different types of shape. That is the secondary structure. If you then take that Lego, and imagine my metaphor is kind of falling apart. Legos aren't flexible. But imagine you had flexible Legos, and you took that string, and you grabbed it, and you bent it. That would be tertiary structure. And if you made 4 of the identical same things, and then stuck those things together, that would be quaternary structure. So the flexing is the folding required for the tertiary structure. So that is proteins. The last type of molecule to talk about are nucleotides. Now, before I even go any further, where have you heard or what do you know about nucleotides and nucleic acids? What are they important in? DNA, yes. So we're going to talk about DNA here. But in order to talk about DNA, we have to talk about the building blocks. Nucleotides, nucleic acids are the building blocks of DNA. So, sorry, I said that wrong. Nucleotides are the building blocks of DNA. DNA is an example of a nucleic acid. DNA is a nucleic acid. It is made out of nucleotides. We only have 5 nucleotides. With proteins, we had 20-some building blocks to work with. Amazingly, for nucleic acids, like DNA, we only have 5. A, T, C, and G. Adenine, thymine, cytosine, guanine, and uracil. The most famous nucleotide, the most common one, the one we're going to talk about over and over and over and over and over again this semester, is the A. A is ATP. This is our energy currency in the body. We burn sugar in our body to make ATP. We then take that ATP, we move it wherever we need energy, and then we use that to get energy right where we need it. I think we're going to see that in a slide or two. Nucleic acids are long strings of nucleotides. Just like we took amino acids and strung them together to make protein, or we took monosaccharides and strung them together to make polysaccharides. If we take nucleotides and string them together, we get nucleic acids. And we only have two different types of nucleic acids, DNA and RNA. DNA is huge. Each DNA molecule contains millions of nucleotides. DNA contains our genes. Our genes are the instructions to make us. They are the blueprints to make us. And we'll see how that process works next week when we talk about the cell. DNA only uses 4 of the 5 nucleotides, A, T, C, and G. It does not use U. RNA uses U, but does not use T. They both use A, C, and G. The DNA also uses T, and RNA uses U. RNA comes in 3 types of its own. We have messenger RNA, ribosomal RNA, and transfer RNA. These do not make sense to you right now. We'll talk about the purpose of those 3 types of RNA next week when we talk about the function of the cell. Now the last thing, how many slides do I have left? So that is all we're going to talk about for nucleic acids at this point. Last thing we want to talk about is some energy in our body. Energy is used to do things in our body, and energy is going to be used by enzymes. Enzymes are proteins, period. Proteins are a chain of amino acids. Some proteins are also enzymes. An enzyme is what I like to call a protein that does something. It doesn't just sit there. It's not like a tent pole holding the cell in place. It is a protein that does something. It has a very specific function. It causes something to happen in the cell. And it does that by lowering what we call the activation energy. And to understand this, we have to go back to our basic chemistry. Now, this isn't a chemistry class, so I'm not going to talk about this too in-depth. But what you're looking at here is called an energy diagram for a chemical reaction. It looks like a roller coaster. We start here, we go up a hill, and then we come down the hill to get to our product. This is why when we heat a reaction, it causes it to go faster. This hill is called the activation energy. In order for 2 reactants to react and make a product, they have to have a certain amount of energy. And the energy required for the reaction to happen is called the activation energy. And so normally, these reactants, in order to form this product, have to get over this large of an energy hill. An enzyme, though, causes the activation energy to be smaller. And so here we see that the hill is smaller. It's much easier for the reactants to get over a small hill than a large hill. By doing that, we're going to allow the reaction to happen quicker. Enzymes are catalysts. They reduce activation energy to cause a reaction to happen very, very quickly. Now, they can't cause a reaction to happen that would not happen on its own. There's always a hill. The thing is, that hill is probably so big that in real life we would never get over it without an enzyme. And so an enzyme comes along, things react, and something happens in our cell. This is an example of an enzyme working. So the green blob is an enzyme. And notice on that enzyme we have two little pockets. We have a U-shaped pocket and a V-shaped pocket. The purpose of this enzyme is to cause this puzzle piece to react with this puzzle piece to form a larger puzzle piece. And these two puzzle pieces happen to fit perfectly into this enzyme. This enzyme exists specifically for this purpose. And so these two substrates, the substrate is the thing the enzyme works on. They're the puzzle pieces. They come in, they stick together. The enzyme causes them to react together to form a product. The product floats away. Now it's important to notice the enzyme was not harmed. It's exactly how it started. An enzyme can be used over and over and over and over again. And remember, this is a protein. This is a very large molecule that our body spent a lot of time and energy into making. And so we don't want it to be a one-time use thing. We want to make it and then be able to use it over and over and over again. Even if we have an enzyme, we have to get over that energy hill, right? Energy in our body is, we said, carried in the form of ATP. ATP stands for adenosine triphosphate. The A is the A, like we saw in the nucleotides. DNA has the A's and RNA has the A's. The triphosphate means that there are 3 phosphate groups. A phosphate group is a chemical entity. It's part of a molecule. But on our diagram here, it is just these P's. And that's really the only level that you need to understand this. We have adenosine, and then we have P's, or phosphates, stuck to it. If we only have 2 P's, or 2 phosphates stuck to adenosine, we have adenosine diphosphate. If we burn glucose, we get some energy from glucose, we can use that energy to stick a 3rd phosphate on. Now we have ATP. And we can take that ATP with its stored energy and move it anywhere in the cell or anywhere in the body that we want energy. And then all we have to do is cut that 3rd phosphate back off. And when we cut the 3rd phosphate back off, we get the energy back out. So that's what we call ATP, the energy currency of the cell. We bring in glucose, nutrients, energy. We use that to get ATP. And once we have ATP, now we can do anything. We can think, we can move, we can digest more food, we can do anything we want to do once we have ATP. So that is the end of our chemistry. Next week we'll talk about cells. Any questions on the chemistry? Anything at all? Okay. If you do have questions later on, certainly just send me a message. I'm happy to help. Again, I suggest that between now and our lecture next week that you do the chemistry the chemistry homework. This next lab, I believe, is a mixture of cells and chemistry. And the chemistry part, actually, you don't even have to do. And so I think you have a break on the lab this week. And so there's no chemistry lab for you to do. So you can wait till next week to do it when you have learned the cells. So before I go, any questions? Okay. Well, so it should be either tonight or tomorrow. I don't know if you noticed on the Canvas homepage, I put a link to a page where after every lecture, I'm just going to add the link for that day's lecture, and so you can go and watch it. So I'll post that link either tonight or tomorrow. So technically, office hours are Thursday at like 9 o'clock or something like that. But that's just because they require me to specifically set aside a time. If we need to discuss something, we can find a time that works for us and get it done. Yeah. So if you have questions, we'll sort it out. You're welcome. Any other questions? Okay. I hope you have a good week. Looks like it's not quite dark. I'm going fishing. Speak now or forever hold your peace. Good night.